Today we're going to talk about cell cycle control, how cells regulate how they proceed through the cell cycle. In controlling the cell cycle, you know that cell size is an important factor. We're talking about surface area to volume ratio. You know you want the surface area to be big and the volume to be small. So you want the biggest possible distance between those two numbers, a big surface area to volume ratio. Refer back to your cell chapter if you don't remember. The surface area will allow material to enter the cell, and the volume, if it's small, will diminish the distance that materials have to travel. This is controlled by the nucleus, and when the cell gets too big, it triggers division. There's other reasons for division other than cell size. The growth and repair of damaged or dead cells, which is mitosis. Reproduction of cells or organisms. In asexual reproduction, we would call it binary fission, or in meiosis, for sexual reproduction, we would call it meiosis. Meiosis is the subject of the next big chapter we're going to work on, so we'll get to that. But regardless, cells can't divide all the time. You know that division has to be controlled, and we've learned in the past 15 or so years that cells division is controlled by checkpoints. And as the cell cycle progresses, G1, S, G2, mitosis, there's checkpoints along the way, and if things are accomplished at those checkpoints, then it can proceed. And if not, then the cell has to back up and do it again. If the repairs are not made, we call it apoptosis, programmed cell suicide. And that means that the cell's lysosomes will actually burst and will digest the cell itself. The cell will digest itself. Programmed cell suicide, apoptosis. Vocab word for you. So here's your cell cycle that you know. G1, S, G2, PMAT, cytokinesis, 2 new cells, started all over again. Please remember that you also have a G0 here, which is a resting phase. However, during the cell cycle, you have checkpoints, and they're indicated by these stop signs. So here's a G1 checkpoint. Apoptosis will occur if, after cytokinesis, in your new daughter cells, the DNA is damaged. And so there's mechanisms for checking this. We'll then proceed through S phase and copy the DNA, go into G2, and towards the end of G2, you have your G2 checkpoint. This is called the mitosis checkpoint because if the cell passes this checkpoint, it will go through mitosis. And what you're looking for is, did the DNA replicate properly? And if not, apoptosis will occur if it can't be fixed. Let's say you pass G1 and you pass G2, then you begin PMAT. So prophase, late prophase, metaphase. In between metaphase and anaphase, you have the M checkpoint. You know that in metaphase, the cell has its chromosomes lined up in the middle, and the spindle fibers are originating from the poles and attached to the chromosomes at the kinetochores and begin to pull the cells apart, the chromosomes apart in anaphase. So what happens is at this M checkpoint, are all the chromosomes lined up in the middle? Is the spindle firmly attached? If yes, then mitosis will continue. If not, then it will try to fix it or force apoptosis yet again. If you pass G1, it does not guarantee that you will complete mitosis. It just guarantees that you continue. Same thing with G2. It just guarantees that you can continue, not necessarily that you can complete the process. So you have to pass all 3 checkpoints in order to completely finish this process. These checkpoints are regulated by what we call cyclins. And cyclins are proteins whose levels rise and fall throughout the cell cycle. And you can Google graph of cyclins, and you'll see that there's several different kinds of cyclins. Each cyclin controls a different checkpoint or even a different aspect of a checkpoint. They call them cyclins because their levels cycle throughout the cell cycle. And the rise and fall of these levels triggers cell division. We're learning more and more about this. We know that cyclins form CDKs or cyclin-dependent kinases, which cause phosphorylation, which triggers different events to occur. But for you, for now, we need to know that cyclins are proteins whose levels rise and fall, and this triggers passage through the cell cycle. One of the other controls on the cell cycle is contact inhibition, and that's basically just what it sounds like. If it touches, it will stop. Okay, so here's a petri dish, and you can see this bottom row of cells. They're all pretty flat. That's good. That's what should happen. This is not good. This is a bunch of cells that didn't exhibit contact inhibition, and they form a mass of cells called a tumor. So contact inhibition should stop division. That's a good thing. But if it doesn't stop division, it can form a tumor, which is a bad thing. A tumor is a mass of dividing cells. The other thing that can control the passage through the cell cycle is external factors like growth factors and hormones. Your nervous and endocrine systems send out signals which tell your cells to divide by bonding to receptor proteins on the cell membrane. When we get to organ systems later in the year, we'll come back and learn about that. Couple final notes on cancer. The definition of cancer is uncontrolled cell division, and a mass of cells is called a tumor. If those cells spread, they're called metastases. It's called a metastatic cancer, or we say that it has metastasized. Cancer can be caused by dysfunctional cyclin genes. Remember, cyclins are proteins, so they have to be made by genes. If those genes don't create the proper cyclin or don't create any cyclin, then that can cause cancer. If you have receptors on your cell membranes that are supposed to receive a signal and cause division, and they're causing division when they shouldn't, that can also cause cancer. Cancer can be considered as inherited, things like breast cancer. The dysfunctional gene actually gets passed down in the gametes from parent to offspring. Most cancers, though, are damaged or caused by genes that are damaged during lifetime by some kind of mutation. You can have a virus. HPV, human papillomavirus, causes cervical cancer. Viruses HPV is caused by cervical cancer. Cervical cancer, all viruses actually insert their DNA into your cells, but some viruses, depending on where it inserts, can actually trigger cancerous changes in the DNA. A lot of common cancers like skin cancer and lung cancer are caused by radiation and chemical exposure, smoking, and sun.